Hi everyone, welcome to MR Learning Circle. I hope everyone is fit and fine and doing good in their lives. So today is the 9th lecture of our MCQ series, and today we are going to cover prokaryotic cells, structure and function. It's the first part, and in this section we are going to complete cell wall, cell membrane and their biosynthesis, as well as mechanism of solute transport across membranes. And I hope you all have watched the previous video. If not, then I'll mention the link above here in the iCard, and I hope you get your copy of your microbiology ebook. You can order, and for ordering you can get the payment link in the description box of the channel. If any doubt, you could contact me, and the email ID is also mentioned there. Any problem, you could get the link on MR Learning Circle Telegram channel. Okay, you just go search MR Learning Circle on Telegram, you'll find the channel and you'll get the link of buying your ebook there. Okay, for any queries, you could email us, you could contact me on Instagram, you can contact me on the email ID given, and all your problems would be solved there. Now let's go ahead. So the first topic which we are going to cover today is cell wall, cell membrane and their biosynthesis. So the first question for this is, which of the following components is unique to bacterial cell walls? Options are A. Chitin, B. Peptidoglycan, C. Cellulose and D is pseudomurine. Correct answer for this question is option B, peptidoglycan. So what is peptidoglycan? It's murin, and it's a polymer of NAG and NAM cross-linked by the peptides, and that it is unique to bacteria and it is absent in archaea and your eukaryotes. Clear? Now let's go to question number 2. Which enzyme catalyzes the final cross-linking of peptidoglycan strands during bacterial cell wall synthesis? Your options are A, MURG, B is transpeptidase, C is transglycosylase and D is autolysin. The correct answer for this question is option B, transpeptidase. So transpeptidase, they are penicillin binding proteins and they form a peptide cross-links between glycan chain in the peptidoglycan. Now next is, which of the following lipids act as a carrier molecule in peptidoglycan biosynthesis? Your options are A. Lipid A, B. Bactoprenol, C is phosphatidyl glycerol and D is cardiolipin. So the correct answer here is also bactoprenol, option B. Question number 4. Lipopolysaccharides are found exclusively in which group of organisms? Options are A. gram-positive bacteria, B. gram-negative bacteria, C. mycoplasma species, D. archaea. Correct option is option B, gram-negative bacteria. Easy question. Now next is question 5. Which antibiotic inhibits the early cytoplasmic step of peptidoglycan synthesis involving the MUR-A enzyme or MUR-A enzyme? Now your options are penicillin, B. vancomycin, C. phosphomycin, D. bacitracin. So the correct answer for this question is option C, phosphomycin. Question 6. Mycoplasma species lack cell wall. What compensates for this deficiency? Okay. A is pseudomurine layer, B. cholesterol in cell membrane, C. S layer protein, D. teichoic acid. So you know B, cholesterol in the cell membrane. So your mycoplasma, they incorporate the cholesterol from the host into their plasma membranes, and that provides the rigidity and the stability in the absence of your peptidoglycan cell wall. Question number 7. Archaeal membranes differ from bacterial membranes primarily in having, your options are A. ester-linked fatty acid, B. ether-linked isoprenoids chain, C is unsaturated phospholipids, D is cholesterol as a stabilizer. Correct answer for this question is option B, ether-linked isoprenoid chain. So archaeal phospholipids, they contain ether linkage between the glycerol and the isoprenoid hydrocarbon chain. Question number 8. Teichoic acids are found in the cell walls of. Options are A. gram-negative bacteria, B. gram-positive bacteria, C. archaea, and D. mycoplasma. Correct answer for this question is option B. gram-positive bacteria. Question 9. Which of the following statements correctly describes the structure of gram-negative bacterial cell envelope? Your options are A. a thick peptidoglycan layer and no outer membrane, B. thin peptidoglycan layer surrounded by an outer membrane containing LPS, C. single lipid bilayer with no peptidoglycan, D is peptidoglycan with teichoic acid. So the correct answer for this question is option B. That is thin peptidoglycan layer surrounded by outer membrane containing LPS. LPS is your lipopolysaccharide. Question 10. Which step in peptidoglycan biosynthesis is inhibited by the antibiotic bacitracin? Your options are polymerization of glycan chain, D. dephosphorylation of bactoprenol carrier, C is cross-linking of peptide, D is synthesis of UDP-NAM. The correct answer for this question is also option B, dephosphorylation of bactoprenol carrier. So this bacitracin blocks the dephosphorylation of bactoprenol that prevents recycling of the lipid carrier. That is now this lipid carrier is required for transporting cell wall precursors across the membrane. So that was for your cell wall, cell membrane and their biosynthesis. Now let's start with mechanism of solutes transport across membranes. First question, which of the following is a feature of passive transport? Options are requires ATP hydrolysis, B. moves solute against their concentration gradient, C. requires specific transport protein but no energy input, D. always involves endocytosis. So the correct answer for this question is option C, requires specific transport proteins but no energy input. Question 2. Which of the following best describes the phosphotransferase enzyme system? That is your PTS in bacteria. Options are A. ATP driven uptake of amino acid, B. proton motive force driven sugar transport, C. group translocation involving phosphorylation of transported sugars, D. facilitated diffusion of ions through channels. Correct answer for this question is option C, group translocation involving phosphorylation of transported groups. Question 3. Which of the following statement about ABC, that is ATP binding cassets, transporters is correct? Options are A. they function only in eukaryotes, B. they require PEP as an energy source, C. they directly utilize ATP hydrolysis for transport, D. they transport solutes only by passive diffusion. Correct answer for this question is option C, they directly utilize ATP hydrolysis for the transport. Question 4. The uptake of lactose by E. coli via the lac permease, that is lac-Y, is an example of. Options are A. primary transport, B. secondary active transport, C. group translocation, D. facilitated diffusion. Correct answer for this question is option B, secondary active transport, that's your symport. Question 5. Which of the following mechanism transports two solutes in opposite direction across the membrane? Options are A. symport, B. uniport, C. antiport, D. facilitated diffusion. Correct answer, as the name signifies, option C, antiport. Question 6. Which bacterial transport system uses a periplasmic binding protein for substrate recognition? Options are A. PTS system, B. simple diffusion, C. ABC transporter, D. ion channel. So the correct answer for this question is option C, that is ABC transporter. Question number 7. The proton motive force used for solute transport consists of which of two components? Options are A. membrane potential and osmotic pressure, B. chemical gradient and electric potential, C. ATP concentration and redox potential, D. proton concentration and water potential. Correct answer option B, that is chemical gradient and electrical potential. Now we have question 8. Which of the following processes allows glycerol to enter E. coli cells? Options are A. Simple diffusion, B. Facilitated diffusion via aquaglycoporin, glyceroporins, C. Active transport using ATP, D. Group translocation. Correct answer for this question is option B. Facilitated diffusion via aquaglyceroporins. Question 9. Sodium potassium ATPase in eukaryotic cell exemplifies. Options are A. Passive transport, B. Group translocation, C. Primary active transport using ATP, D. Secondary active transport driven by PMF. Correct answer for this question is option C. Primary active transport using ATP. Question number 10. Which of the following correctly pairs the transport type with its energy source? Options are A. Facilitated diffusion, ATP hydrolysis, B. ABC transporter, that is your proton motive force, PTS, phosphoenol pyruvate, D is light energy. The correct answer for this question is option C. PTS system and phosphoenol pyruvate. So that was all for the lecture. Thank you for watching, and if you have learned anything from this lecture, please do like, share and comment down below, and study well. Thanks for watching.